Hi, my name is Deja Tillman. Today is March 17th, 2020, and I am in exercise 1, culturing microbes and broth. Okay? This is week 1, once again. So with this lab, I have my PPE on, goggles, gloves, apron, long sleeves, and closed-toed shoes. I already wiped my surface down with 10% bleach. I have 3 vials of the nutrient broth. I'll set it up right here so you guys can see. This one is E. coli. This is S. epidermidis. And the last one is the yeast lactobacilli. The yeast, I have it here. And the E. coli is in this bottle here. And the S. epidermidis is here. So basically what I'm going to do is get these inactive microbes, put them in the broth with the labels, and monitor them 48 hours and keep them in incubation to observe micro growth pretty much. So I'm going to get started. I'm going to start with the E. coli broth that's labeled. Twist the tip off. I'm lighting my candle now. I will be putting the tip of the vial around the flame to sterilize it. Just like that. And with the E. coli, I will be doing the same thing. Just the tip, sterilizing it for aseptic technique. It's hard since the vials are small, so be careful. Don't burn the gloves off. Alrighty. I'll leave this here. I have my pipette here filled with alcohol that keeps it sterile. I'm going to shake it, make sure all the alcohol is out. Okay. All the alcohol is out. Nice and dry. And I'm going to grab once again, the broth labeled E. coli. And then I am going to get a small sample. Alcohol is off, it's dry. And I'm going to be getting this pipette, drawing 0.25. Right there, perfect. That's good right there. I'll place that back down. I get my E. coli. Be careful it doesn't touch. And just pour the juice in. I'm going to put this back in alcohol, recap the E. coli, and shake it. So mix it. You have to shake it vigorously so it can dissolve. Perfect. Looks like it's dissolving here. Perfect. Very good. Now there's bubbles. I don't want to shake it too much. Perfect. Nice and mixed together. So as you guys can see, I'm going to move the fire over for safety just because I'm wearing plastic and I don't want the plastic to catch on fire. My lighter, I'll put it over here. So pretty much it is partially dissolved. I'm going to shake it a little bit more, you guys. I'm sorry about this. Okay, I'm going to uncap it. Make sure I'm not touching the tip of the vial. And with this pipette, nice and clean, I'm going to reinsert it. Make sure the tip is not touching and try to get all that fluid out, which I have successfully gotten out. Alrighty. I'm trying to get all my fluids in here. Sorry, guys. There we go. Then we are gonna put it into the broth. Perfect. Re-sterilize the pipette. Burn it off to sterilize the top of the cap. A little bit more. Put the cap back on and keep it off to the side. Perfect. Put my cap back on for safety. We're going to repeat the steps for the S epidermis. I put all the trash to the side. So we're going to do the S epidermis, which is this one labeled S epidermis. We're going to sterilize the tip of the tube. Nice and sterilized. You're going to spin, spin, spin. Same with the S epidermis here. And re-sterilize it. Okay, nice and sterilized. So I'm going to clean my pipette. Sorry. Okay, all the alcohol is out. I'm going to sterilely place some in here. 0.25 mLs. Then put it with the S epidermis. Perfect. Pull it out, put it back in the alcohol to keep it sterile. Put the top back on, and we're going to shake once again. Shake once again. Make sure it is completely dissolved. That is kind of thick, so I'm going to keep shaking it. We're shaking and shaking. I have all my pipettes so I don't drop anything. I put it back in this. Alrighty, guys. Almost dissolved. We're going to shake it a little bit more. Okay. There's a little tiny pieces of chunks in there. Still going to shake it. Okay, so we should be good. Take this off. Take the top off. Rinse the pipette. Nice and sterile, nice and clean. Then we're going to enter it carefully. Don't touch the tip of it. Get everything out. And drop it into the labeling broth of the S epidermis. Perfect. If you can get the rest of it, but just make sure the pipette does not touch. Perfect. Put the pipette back in the alcohol. Put the top back on the S epidermis for safety. The cap is gonna, before you put it back on, you're sterilizing it. Perfect. Put the top back on, and that's it. So we have warm tap water mixed with a half a teaspoon of yeast. So I'm putting the yeast in. A little bit of the yeast and warm tap water, which I can put in now. And we're going to do the same exact thing that we've been doing with the other test experiments. Water is warm. Perfect. And then we're going to grab 0.25 of the yeast from the pipette. Perfect. And then we'll want to mix, mix, mix. And it's nice and mixed. We then are going to sterilize the top. Get the pipette. Make sure the pipette's clean. Grab 0.25 of the yeast. Perfect. Then you're gonna put the yeast inside the broth, labeled yeast, E. coli. Okay. Serobasis. Perfect. And then after that, once all 3 broths are completely full, you are going to let it sit for 48 hours, and then we're just going to watch the yeast grow. So I'm extremely excited for this experiment, and I'm excited to look under the microscope to see what's going on. Alrighty, guys, thank you so much for being patient with me and watching this video, and I bless you guys. Hope you guys have a wonderful day. Bye-bye. Thank you. Once again, sorry, March 17th today.